Arbuscular mycorrhizae form symbiosis with the roots of over 80% of plant species, including ferns, grasses, trees, and almost all of our most important crops. These species grow asexually in the soil. Plant roots send out strigolactones, which induce the fungi to branch and grow further, and the fungi send out molecules which activate root genes to allow them to colonize. The fungal hyphae penetrate the root's epidermis, grow between the cells, and then form arbuscules within the cells. These arbuscules are where the fungi exchange phosphorus and other nutrients from the soil for carbon which the plants have fixed from the atmosphere. This was especially important for early land plants as they did not have roots to obtain scarce nutrients from the soil. In fact, arbuscules are found in the roots of 400 million-year-old plant fossils. Combined with the fact that almost all plant families have this symbiosis, and that the fungi which associate with the plants are monophyletic, suggest a single ancient origin for this interaction. Gene sequences from plants which allow fungal colonization are highly conserved across plant families in terms of both base sequence and protein structure, suggesting high selection pressure and a big fitness benefit. Understanding this symbiosis is important to improving soil health and fertilizer use in agriculture, as well as understanding the globe and carbon cycle, as mycorrhizae form an important part in carbon sequestration.